So welcome back from the break. Let's take a look at our concept check question, and then we'll start talking about DNA replication and how the process proceeds. So compare and contrast the structures of the DNA and RNA molecule. Well, compare means what are the similarities? So the similarity is that they are built out of 4 nucleotides. Each of them contains 4 nucleotides. Those nucleotides have the same basic format, a sugar, a 5-carbon sugar, with a nitrogenous base attached to the 1' of the sugar and a phosphate group attached to the 5' of the sugar. There the similarities end. What are the 3 differences? Well, the 3 differences are that DNA is a double-stranded polynucleotide, so it consists of 2 strands of polynucleotides, and RNA is a single-stranded polynucleotide. The sugar in the nucleotides for DNA is deoxyribose, hence the name deoxyribose nucleic acid, and the sugar in the RNA nucleotides is ribose, hence the name ribose nucleic acid. And then, of course, while they may be formed from 4 nucleotides, it's not the same nitrogenous bases in those 4 nucleotides. The A, G, and C nucleotides are the same. So adenine, guanine, and cytosine nucleotides are present in both DNA and RNA. But in DNA, we have the thymine nucleotide, whereas in RNA, we have the uracil nucleotide. So those are the compare and contrast things that you should know about the DNA molecule. So let's go on and talk about DNA replication. So as I said in the previous section, Watson and Crick's model clearly indicates that semi-conservative replication is probably the best way for DNA to be replicated, and that's what they suggested. As we now know, that is in fact the way it is done. And the Messelson-Stahl experiment that was published in 1956 clearly indicated that it is semi-conservative, as opposed to 2 other alternatives that could potentially be used, one which was referred to as conservative, meaning that the original DNA molecule was fully conserved and any new DNA molecules formed were totally new. And the other was referred to as dispersive, where there was no attempt to maintain the integrity of either of the 2 strands of the original DNA molecule, and somehow the product of replication, the DNA molecules as a product of the replication, represented a jigsaw puzzle or mishmash of the original DNA molecule and newly generated DNA. Now, Messelson and Stahl did not try and explain how the individual replication processes took place. All they were wanting to show was, is it semi-conservative or not? And so their paper focused on trying to differentiate semi-conservative from the other 2, and they were able to successfully show that semi-conservative is the way in which DNA is replicated. So if you want to see a very nice animation of the process as we understand it today, taking into account everything that we know about the DNA replication machinery, because it really is a machine, this animation is really, really good. It's based on an even more complicated animation that comes from the Howard Hughes Medical Institute, HHMI. I do have a copy of that animation, and I've hesitated to post it simply because of the degree of detail and complexity that it gets into. This is probably a better one for you to take a look at. But even this one gets a little bit complicated in terms of telling you how everything takes place. But of course, you are working on an infographic, and hopefully you've started working on that covers replication, transcription, and translation, the topics of this chapter. So that will help you understand the process as you do your own research. So we're going to break this down into sections to show how DNA replication actually takes place. And there's a series of animations, simpler animations, that walk you through the various stages of DNA replication. So this is a simpler animation showing the semi-conservative replication process. This is a simple animation of the semi-conservative replication. So after you've looked at all the detail in the BioFlix animation, you can come and take a look at this one and see it laid out in a very simple fashion that makes it a whole lot easier to understand what's going on. So this animation, again, is a little bit more complicated, but it takes an overview of the whole of the DNA replication process. So this is, I'll just put in here a big picture. What happens as we go through the next part of this presentation is to take a look at individual sections of this animation, starting with what you'll discover here, something called the origin of replication, or the replication bubble, and then the individual replication of the two strands known as the leading strand and the lagging strand, and also the priming process that gets everything started on the strands. And it also identifies some of the enzymes that are involved. All these are pieces of information that you need to have in place as you come to understand the process of DNA replication. So let's start breaking it down into smaller pieces. So this diagram basically tells you what's going on. The existing DNA is unzipped. And that is, in fact, the technical term that we use for this. We talk about the DNA molecule being unzipped because it's opened up in much the same way as a zipper is open. It looks very much like that. And two existing strands. So we build the complementary strands using the base pairing rules. And you should remember what they are. A pairs with T, G pairs with C. Now, the added complexity here is due to the fact that the two strands are anti-parallel. So one of the things that we can never forget is the fact that the two strands are anti-parallel. That affects a whole lot of things, as you'll see. So the enzymes that actually do the copying are called DNA polymerases, and they have a very specific feature that actually adds to this complexity of wanting to do the two strands that are anti-parallel to each other at the same time. So a DNA polymerase can only add nucleotides to the 3' end. So this is one of the limitations of a DNA polymerase. It only knows how to add nucleotides to the 3' end of the growing polynucleotide strand that it's creating. But it also has another drawback. It can't start copying a single-stranded DNA molecule or a single polynucleotide strand. It needs a short sequence of double-stranded nucleotides in order to be able to start adding new nucleotides to the existing 3' end of that short piece of double-stranded nucleotides, as you'll see. I'll show you exactly how that happens along the way. What we also know is that DNA replication does not start just anywhere. There are specific locations, we refer to them as origins of replication, where the two strands are going to be separated from each other and stabilized so that they won't spontaneously link back together again, which they otherwise would have if they weren't stabilized. And then enzymes, which we refer to as helicases or helicases, start to unwind and separate the two strands, creating what is referred to as a replication bubble. And the helicases actually unwind and separate the two strands in both directions. So they actually move away from the origin of replication going in both directions. So the replication bubble grows and expands, moving away in both directions away from the origin of replication. And replication occurs at the point where the two strands are opening up, what's referred to as a replication fork at the ends of the bubble, at the opposite ends of the bubble. That's where all the action is taking place. So what we want to focus on is first, what happens at the origin of replication? How is DNA replication triggered once the origin of replication opens up and the two strands are separated from each other? And then what is the ongoing process of replicating the two strands, given that one is anti-parallel to the other? What effect does that have on the replication of the two strands? Because it's got to have some effect. So this first animation details the origin of replication. What is the function of the origin of replication? What happens at the origin of replication? How does the replication bubble actually get generated from the origin of replication? So it shows the origin of replication forming, the two strands are opened up, and then it gives you a quick look at the role of the helicase as the unwinding and unzipping enzyme functioning at each end of the replication bubble. The next animation details Not on the lagging strand, it's on the leading strand. Oops. That's going to fit harder. Let's put it in leading strand. There we go. So the way, remember again, that the DNA polymerase cannot start copying a single-stranded polynucleotide strand. It simply doesn't know how to do that. And so in order to start the copying process, a short sequence of RNA nucleotides is added in by this enzyme called primase on the existing template strand. And because it's now a short section of double-stranded nucleotides, the DNA polymerase knows what to do. It binds onto the single-stranded DNA adjacent to the primer, that's the name that we give to this short section of RNA nucleotides, and adjacent to the 3' end of that primer, and starts adding in DNA nucleotides on the basis of what the nucleotides are in the template strand. And that strand that is generated in that fashion is called the leading strand. So there are two rules that you need to recognize that come into play here. And this is consistent wherever DNA is being read and wherever DNA is being copied. So when we talk about transcription, these same rules are going to apply in transcription as well. So DNA templates are always read 5' to 3'. So when a DNA polymerase or the enzyme in transcription called RNA polymerase reads the template strand, it's always reading that strand in the 5' to 3' direction. But the new strand is, oops, I never remember that I've got these things coming in. Put it here. I think I can do it. This is a nuisance sometimes. I don't want to see those. I'd like them to go away and stay away. Okay, we'll put it here, created in the 5' to 3' direction. Okay, so the DNA template is always read 5' to 3', but the new strand that's being built as a consequence of copying that information is always going to be created 5' to 3'. And actually, I've got a mistake here. So let's correct that. The DNA is always read 3' to 5'. I knew there was something wrong there. So the template strand is always being read 3' to 5'. But the new strand that's being created by the polymerase, whether it be a DNA polymerase or an RNA polymerase, is always going to be assembled in the 5' to 3' direction. Those are hard and fast rules. And that's consistent irrespective of where we're going with this when we're talking about the replication. So the other thing that I want to do is define the leading strand, because that's what this animation is actually referencing, the leading strand. Primed once, then continuously copied. Actually, we should say replicated, not copied. So again, the template is red 3' to 5'. So the leading strand only ever gets primed once at the origin of replication, and then as the replication bubble opens up and the replication fork gets created, the DNA polymerase starts reading that strand 3' to 5' and just continuously copies it as the fork opens up. So that's the leading strand. The other strand is called the lagging strand. So the lagging strand is continuously reprimed. And copied in chunks called Okazaki fragments. So this is where the added complexity comes in, because you are reading 3' to 5' in this direction, and then you're building 5' to 3'. It stands to reason then that you are reading this one 3' to 5' in this direction. So this is the reason why the lagging strand has to be done in a slightly different way. It's still mostly the same enzymes, but there are a couple of extra enzymes that are involved. So it's still primase that puts the primer in. It's still the same DNA polymerase that does the initial copying. But that polymerase does not know how to remove the primer RNA nucleotides. So if it starts here and reads down in this and copies down in this direction, when it gets to this primer, it stops because it doesn't know what to do with this. But fortunately, there is a second DNA polymerase. In bacteria, there are 5 DNA polymerases in total. In eukaryotes, there are 4. And each of them has a different role in copying DNA and repairing damage to DNA. So when the initial DNA polymerase that does the copying gets here, it stops and it simply disengages from the DNA. But a second DNA polymerase then comes in, removes the RNA nucleotides and replaces them with the appropriate DNA nucleotides. But that still doesn't finish the process, because there's always going to be a gap between this fragment and this fragment, because the DNA polymerases simply can't fill that gap. Remember, again, they can only add nucleotides to 3' ends. They can't fill gaps. So fortunately, there is a third enzyme called DNA ligase, and this is detailed here in the animation, that can fill in the gaps. So in order to do the lagging strands, you need a couple of extra enzymes to take care of that added complexity. You can't just prime it once and copy it continuously. You have to constantly keep repriming it. But that creates the problem because the DNA polymerase that's doing the copying does not know how to deal with the RNA nucleotides of the primer. Fortunately, a second DNA polymerase does. So that second DNA polymerase takes care of the primer nucleotides. But we still have a problem in that the polymerases can never seal gaps between the fragments. So a third enzyme, the DNA ligase, fills in the gaps. Adds some extra complexity to the whole process, but nonetheless, it gets the job done. And what is interesting here is that labeling these leading and lagging strand might imply that the lagging strand is copied a little bit slower. And that would be inaccurate because it turns out that both strands are copied at essentially the same rate, even though there's more involved in copying the lagging strand than the leading strand. Yeah, I should write across the whole of the bottom there, I guess. I'll learn. Okay, so again, this animation is in the resource page. All of these animations are in the resources page. The big picture one that covers everything, and then all of these small pieces taken out of that, focusing on the different parts of it. They're all in the resource page, so you can take a look at these in the resource page. In the end, this again is a pretty simplistic diagram, but it illustrates again what is basically going on. You start with the chromosomal DNA. There are multiple origins of replication on the chromosomes of a eukaryote. There could be upwards of 100 or more origins of replication on some of the bigger chromosomes. Each one of them activates at essentially the same time, opening up their own unique replication bubbles. The bubbles expand as helicase unwinds the DNA, and obviously the bubbles then start to expand in both directions. The leading strand and the lagging strand at each replication fork are replicated at the same time as the bubble expands, and the bubbles grow towards each other. Eventually, the bubbles merge. The fragments of on each strand are linked together finally by a DNA ligase enzyme, the same one that was linking the fragments together on the lagging strand. It finally links all the pieces together. And eventually, as all the bubbles merge and all the fragments get linked together, you wind up with two daughter DNA molecules. And these would be the two sister chromatids. So these would become the two sister chromatids of a replicated chromosome or duplicated chromosome during cell division, whether it be mitosis or meiosis. So now we have an explanation of where those sister chromatids actually come from. They are the product of this replication process. So DNA replication is important because it makes sure that all the body cells have a complete set of chromosomes. And that was the whole point of replicating the DNA before mitosis, that all the daughter cells should have a complete set of chromosomes. And also in meiosis, having replicated the chromosomes, it means that the gametes have copies of the chromosomes to pass on to the next generation. So DNA replication is obviously a very, very important process. So, time for another break. We are next going to go and talk about transcription, the process of starting to extract information out of the DNA molecule. But before that, let's take a quick break and a concept check. Compare the replication of the leading strand and the lagging strand of the DNA molecule. Identify the different enzymes involved and their roles. See you on the other side of the break.